As we begin our considerations of the process of DNA replication, let's first look at the general mechanism and then look at the experimental evidence that proves it. We can imagine several challenges to the process of replication. Since the DNA is a supercoiled double helix, we'll need to unwind and separate the two strands. Because the helix forms spontaneously, this will not be an energetically favorable process. Next, we recognize that the strands are in an anti-parallel orientation, so that will also need to be taken into account. Since maintaining fidelity of the message is essential, there must be some way of guarding against nucleotide incorporation errors. We might imagine different mechanisms for replication, but we find the process is semi-conservative in nature. That is to say, each original or parent strand is used as a template to synthesize a new or daughter strand. Thus, the resulting two copies each carries one parent and one daughter strand. This means that one half of the original DNA is retained in the new double helices. It is semi-conserved, as shown here in our figure. Let's take a look at the experimental evidence that proves this mechanism. Cultures of the bacterium E. coli were grown in the presence of ammonium chloride carrying an N15 isotope. This means that any newly synthesized nitrogen-containing compound would carry the heavier N15 label rather than the lighter, naturally occurring N14 label. Allowing for growth and full incorporation of the N15 label, the cells are then transferred to a medium with N14 labeled ammonium chloride. The DNA is then extracted from cells for analysis. As in every good experiment, controls must be included. In this case, including cells grown only in N15 or N14 media. The DNA samples were then subjected to density gradient centrifugation. Molecules will sediment within this gradient based on mass or molecular weight, with heavier molecules moving toward the bottom of the centrifuge tube. In the figure shown here, we can see the purpose of our N14 only sample on the left and the N15 only sample on the right. These serve as reference points for the migration of our test samples. In the second tube from the left, we see the results of the first generation of N15 incorporation. Note that the sample migrates to a molecular weight that is halfway between that of the N14 and N15 samples, proving that the new DNA was composed of one strand containing N14 labeled nucleotides and one strand containing N15 labeled nucleotides. In the second generation tube, after transfer to N14 medium, we see two species, the N15 parent in combination with an N14 daughter, and the N14 parent in combination with an N14 daughter. These results will serve as an excellent example of how a properly designed and executed experimental protocol can be used to prove or disprove a given theory.